We go to question number 65. During replication of the bacterial chromosome, DNA synthesis starts from the replication origin and choices are A. RNA primers are involved, B. is facilitated by telomerase, C. moves in one direction of the site, D. moves in bidirectional way. Now, when you take bacterial DNA, the bacterial DNA is circular. So, it is a circular bacterial DNA and it has one point of origin. It has an ORI. During bacterial DNA synthesis, which has been studied by Keynes and often called as the rolling coil model, new strands are synthesized in such a way that from the point of origin it moves on either sides. So, this synthesis of new strands on either sides, which is facilitated by an origin O, and the strand synthesis occurs like this. This synthesis is termed as bidirectional synthesis. Hence, the answer is D. We go to question number 66. In transgenics, expression of transgene in target tissues is determined by A. enhancer, B. transgene, C. promoter, D is reporter. Now, when you take transgenic crops, in a transgenic crop, you have a transgene. Transgene is a foreign gene of insert which is flanked, which is a foreign gene of insert. It has a right flank and it has a left flank. The foreign gene of insert can express gene expression in a target tissue. This is the target cell or the target tissue. When you primarily say expression, gene expression means DNA making RNA and RNA making proteins. This primarily is the meaning of the term gene expression. So, always expression is controlled by the gene itself. The gene itself. That is, the transgene controls the gene expression, whereas an enhancer is a sort of a gene which can improve or increase the efficiency of gene expression twice. Enhancer, like in case you have a gene called A double A is called enhancer. A single gene increasing the dosage of the expression of the gene is enhancer. A promoter is one which improves the efficiency. Supposing we have a gene here and you want to improve the gene efficiency, you want to go it on a faster run. within a given period of time, how much of the product of the gene is improved. That is primarily determined by the promoter. So, promoter is related to efficiency. Whereas a reporter is primarily, you got in plants, you got GUS reporter. A reporter, like you take a green dye protein, if the transgenic program, if the gene expression program is completed, or if the gene is expressed, then the report that it is expressed is determined by another gene which is called a reporter gene, which normally is a sort of a dye gene. It is tagged with a green dye. Thereby, when you take normal transgenic crops, when you are looking at expression, gene expression in the target tissue is predominantly determined by the target gene. If you take plants, in plants you got the expression of gene for phosphoenol pyruvate carboxylase, which is the sort of a gene which expresses the C4 photosynthesis. Similarly, you got even the gene for ribulose diphosphate carboxylase in photosynthesis, which occurs in C3 crops. Then again, you got NIF genes, nitrogen fixing genes in legumes, and you got even nodulating genes in legumes. In case we take a nitrogen fixing crop and a gene and try to express it in some other crop by genetic engineering, if we want to know whether NIF is expressed or NOD is expressed, or gene coding for PEP carboxylase or ribulose diphosphate carboxylase is expressed, then it primarily is the expression of the transgenic crop. Hence, the answer is B. We go to question number 67. During transcription, holoenzyme RNA polymerase binds to the DNA sequence and the DNA assumes a saddle-like structure at that point. What is that sequence called? Choice A is AAAT sequence, B is Tada box, C GGTT box, D is CAAT box. Now, whenever there is transcription, during transcription, we understand that this is the DNA. This is the DNA, and we have an RNA polymerase sequence, and the RNA polymerase sequence forms a saddle-like structure. This is a saddle-like structure or configuration which is formed on the left away from the point of origin, and the sequence of nitrogen bases which is recognized by the RNA polymerase, this sequence is termed as TATA box, Tada box. So, Tada box is a sequence whereby there is more of TA and more of TA. So, Tada box is otherwise termed as consensus sequence. It is termed as the first consensus sequence which is required during transcription in the case of eukaryotes. The answer is B. Question number 68. Which one of the following makes use of RNA as a template to synthesize DNA? We have choices as A is DNA polymerase, B is RNA polymerase, C is reverse transcriptase, D is DNA dependent RNA polymerase. The question asks which one of the following makes use of RNA, like which is the sort of an enzyme, which makes use of RNA as a template to synthesize a DNA. As you understand, choice A, DNA polymerase. DNA polymerase is primarily involved in protein synthesis. When you take RNA polymerase is involved with relation to synthesis of the RNA. Whereas DNA dependent RNA polymerase, choice number D, is used for synthesis of RNA from DNA. DNA in the presence of DNA dependent RNA polymerase makes an RNA. Whereas if you use an RNA template to form a DNA, it occurs in the presence of an enzyme called reverse transcriptase. Because the entire process of transcription is reversed, which occurs in cancerous cells, it is otherwise termed as teminism. So normally you have the DNA making the RNA, which is called transcription, whereas in the presence of a reverse transcription enzyme, the opposite is true. RNA acts like the template and it synthesizes a DNA strand. The answer is C, reverse transcriptase. We go to question number 69. Telomerase is an enzyme which is, choice A is a simple protein, B RNA, C is ribonucleoprotein, D it is a repetitive DNA. When you take telomerase, now telomerase primarily is a molecule which is rich in RNA, and so it is an RNA binding enzyme, so you call it as a RNA enzyme. It is associated with chromosomes. It is associated with the tip of the chromosome called a telomere. When you take a chromosome, this part is termed as a centromere. The arm is termed as a chromatid. You have a pair of homologous chromosomes coming in. Both of them constitute a homologous pair. The tip of the chromosome is called a telomere, and the telomere has the telomerase. And telomerase, because it is made up of RNA, and primarily it is made up of ribonucleoprotein. So it is both a protein component as well as it is also integrated with the ribose. Hence the answer is C. We go to question number 70. The one gene, one enzyme hypothesis was postulated by A. Hershey and Chase, B. Gerard, C. Beadle and Totem, D is Franklin. When you take the one gene, one enzyme hypothesis, The one gene, one enzyme hypothesis has been proposed by Beadle and Totem. Who had worked with the fungus called Neurospora crassa. This area of working relates to the study of haploid genetics. They found out when you take the fungus, the fungi have got spores. The spores are haploid in number and the spores could be mutated. So, you can have the mutant spores grown in a petri dish. The mutant spores can be grown in a petri dish. So, they found out that for every particular gene which is present on the DNA, this gene is responsible for the synthesis of one single enzyme. So, it is one gene and one enzyme, and this sort of a this was the first evidence to indicate the link between genetics and biochemistry, and it comes in the area of biochemical genetics. And it involved the study of mutations. Whereas if you take choice A, Hershey and Chase, Hershey and Chase gave the first direct evidence that DNA is genetic material. Gerard is involved with relation to study of blood groups, and Franklin is associated with X-ray diffraction crystallization of the DNA. The correct answer is Beadle and Totem, that is answer is C. Question number 71. While doing the multiple choice, along with solving the choice of the multiples, we are also going into the concept base and looking at the background. So, when you come to question number 71, in which mode of inheritance do you expect more paternal influence among the offsprings? When you take the modes of inheritance, we have the Mendelian inheritance, as well as we also have the non-Mendelian inheritance. Later on, we also have variations of the Mendelian inheritance in terms of gene interaction, in terms of cytoplasmic inheritance, in terms of multiple alleles, etc. Now, we are looking at the mode of inheritance. In which mode of inheritance do you expect more maternal influence among the offsprings? The choices are: A is cytoplasmic, B is Y-linked, C is X-linked, D is autosomal. When you take autosomes, autosomes are sex chromosomes, and when you take X or Y, it is sex limited chromosomes. Whereas whenever we have traits like cytoplasmic male sterility, CMS, like we have cytoplasmic male sterility, or we have the inheritance of chloroplasts, or we look at variegation in plants. Variegation in plants is sometimes if you take crotons. In crotons, we have the leaves. In the leaves, we have yellow spots, green spots and white spots which are formed on the leaves. They are like a mosaic pattern. So, these patterns are primarily due to inheritance of chloroplast. Now, chloroplasts are always present in the chloroplast. When you take chloroplasts are organelles which have got the chloroplast DNA. Similarly, you got mitochondrial DNA in the mitochondria. Both chloroplast and mitochondria are present in the cytoplasm. Now, when you take the cytoplasm, we have the nucleus and as well as the cytoplasm in a plant cell. When you take the cytoplasm, all characters which are inherited through the cytoplasm, that is cytoplasmic inheritance, is otherwise termed as maternal inheritance. That is through the mother or through the egg. It is primarily through the egg, that is the ovary, ovule and the egg, maternal inheritance, and it is otherwise termed as cytoplasmic inheritance or extra chromosomal inheritance. Is otherwise termed as extra chromosomal inheritance. So, more of maternal influence occurs in the offsprings primarily due to cytoplasmic inheritance, that is choice is A. The answer is A. Question number 72. The Okazaki fragments in the DNA chain growth, choices are A. polymerize in the 3' to 5' direction and forms the replication fork. B. prove semi-conservative nature of DNA replication. C. polymerize in the 5' to the 3' direction and explain 3 to 5' direction of DNA replication. D is result of transcription. So, the question asks here about Okazaki fragments and how do they grow in the DNA chain. We understand that during DNA replication, there is the formation of a replication fork. The replication fork always proceeds in this direction, that is from the right to the left. So, primarily here, when you take the 5' direction to the 3' direction, from the 5' to the 3' direction, when small pieces of DNA are joined, these DNA pieces are termed as Okazaki fragments. So, the Okazaki fragments polymerize in the 5' 3' direction. The word here is polymerize. That is, they join due to the presence of a DNA ligase, and they also explain the 3' to 5' DNA replication. They explain how the replication occurs in the 3' to the 5' direction. We have the formation of a lag strand and also the formation of a lead strand. So, Okazaki fragments give evidences for DNA polymerization and also the process of DNA replication. The answer is C. Question number 73. The length of the DNA molecule greatly exceeds the dimensions of the nucleus in eukaryotic cell. When you take the length of the DNA is just like a noodle and it can run to about 5 meters in length. How can such a big DNA get accommodated within a normal cell? How is this DNA accommodated? A is supercoiling in nucleosomes, B DNA digestion, C through elimination of repetitive DNA, D deletion of non-essential genes. When you have a large length of the DNA molecule, the large length of the DNA molecule is wound round structures called nucleosomes. So, nucleosome is a protein complex. It is a protein and it is rich in histones. So, the DNA is bound two times, and again there is a DNA linker. The linker again, and it joins another sort of a nucleosome. So, this is nucleosome 1, this is nucleosome 2, and the DNA acts like a sort of a linker. Now, when you take the entire DNA molecule, these nucleosomes in turn, they supercoil. That is, all the nucleosomes come together and supercoil, just like if you on a string you have 200 matchboxes, all the matchboxes you again round them up. So, this method of coiling, DNA getting coiled around the nucleosome and nucleosome getting coiled to form a sort of a supercoil. Supercoil is termed as the nucleosome model of packing. Which has been studied by Korenberg. The answer primarily, so you accommodate the entire length of the DNA by supercoiling in nucleosomes. The choice answer is A. While if you take B, choice B is DNase digestion. If you are using an enzyme called DNase, you break the DNA into small pieces, again the DNA pieces will not join initially. Choice C, through elimination of repetitive DNA, no editing occurs during DNA packing. And D, deletion of non-essential genes. Always non-essential genes are also important in the DNA molecule, because it is just like a sort of a toolbox from which whenever it is needed in case of a repair or in case of a damage, a nitrogen base is always taken from the non-essential region and again used in the essential region. Hence, the right answer is A, that is the supercoiling modeling, that is supercoiling in the case of nucleosomes. Question number 74. A sequential expression of a set of human genes is A, messenger RNA, B, DNA sequence, C, ribosomes, D is transfer RNA. Whenever you have genes and you are looking at sequential, and again you are looking at expression, that is, we are dealing with gene expression. Gene expression, which is sequential, is determined by the DNA sequence. A DNA sequence of codons. A codon is a set of 3 successive nucleotides is termed as a codon. The DNA sequence dictates the sequence on the mRNA, which in turn translates into a sequence on the tRNA. The tRNA has the anticodon. The anticodon is specific to an amino acid. So, an amino acid, when it combines with the tRNA with an anticodon, the tRNA remains charged. The charged tRNA in turn recognizes an mRNA, which is formed from a codon sequence of the DNA. Hence, the key to the question is the DNA sequence. The answer is B. Question number 75. during transcription, the RNA polymerase holoenzyme binds to a gene promoter and assumes a saddle-like structure. What is the DNA binding sequence called? The choices are: A is AATT, B is CACC, C is TATA, and choice D is TTAA. Now, whenever an RNA polymerase binds, when it binds to a promoter sequence. It binds to a promoter sequence, and when the RNA binds to the promoter sequence, it forms a saddle-like sequence. On binding, it forms a saddle-like sequence. And in the saddle-like sequence, when here on the promoter you are looking at the binding site. This is called the binding site. The binding site, the DNA binding site, as implied, has a consensus sequence. So, the DNA binding site or the consensus sequence, due to which you get a saddle-like configuration, is termed as the Tata box. Is termed as a Tata box. The answer is C. Question number 76. For the first molecule which can code, what is needed? A. CHONSP, B. CN and P, C. CNOP, D. None of the above. Now, if we have an organism and the organism need code, the first molecule which the organism need to have is its nucleic acid. And all nucleic acids are predominantly derived from the elements of carbon, which is a component of the sucrose or the glucose molecule. Then it has hydrogen. Hydrogen is a very important molecule because it forms hydrogen bonding. Oxygen is a basic requirement of all nucleic acids. Then nucleic acids by nature are made up of the element nitrogen too, and sometimes even sulfur as well as phosphorus. So, in order that an organism primarily structures its nucleic acid, which is made up of a nucleotide or a nucleoside, and the basic molecules being the sucrose, the nitrogen base, as well as the van der Waals forces, as well as the hydrogen bonds, the water bonding, hydrogen bonding, and again due to the presence of the purines and the pyrimidines, the basic structure of the purines and the pyrimidines are made up of CHONSP. So, in order it codes, first a DNA has to get formed with the formation of the nucleotides and the nucleosides, and also the sugar phosphate linkages. Thereby, among the choices, the organism which can code need to have all the 6 elements. Hence, the answer is A, that includes CHONSP. Thereby, even if we were to discover the most primitive organism which has existed or which is existing in outside planet Earth, that particular organism should have the basic elements of CHONSP in order to primarily do the function of genetic code. So, genetic code is basically a most important fundamental property of life processes. Question number 77. One gene, one enzyme relationship was established for the first time in A is Salmonella typhimurium, B. Escherichia coli, C. Escherichia coli, D. Neurospora crassa. The right answer is D. Neurospora crassa. Question number 78. In the DNA molecule, the choices are A. The total amount of purine nucleotides and pyrimidine nucleotides is not always equal. B. There are two strands which run parallel in the 5' to 3' direction. C. The proportion of adenine in relation to thymine varies with the organism. D. There are two strands which run anti-parallel, one in the 5' 3' direction and the other in the 3' 5' direction. When you take a DNA molecule, the basic property of the DNA molecule is it shows opposite polarity. When you understand by opposite polarity, we understand that one of the strands runs in the 3' 5' direction, whereas the other strand runs in the opposite direction, that is 3' 5' direction. Thereby, there are two strands showing opposite polarity, and because it shows opposite polarity, this is the basis for complementary base pairing. Choice A says total amount of purines and pyrimidines is not always equal. It is always equal. The choice A gives a negative statement. B, both the strands run parallel to 5' 3' direction is wrong because it should show opposite polarity. And again C, the proportion of adenine in relation to thymine varies with the organism. It does not vary with an organism. It is constant for an organism. Hence, the right answer is D. Question number 79. Which one of the following pairs of codons is correctly matched with their function or the signal for a particular amino acid? The choice A says GUU, GCU for alanine, B UAG, UGA as stop codons, C AUG, ACG as start and also coding for methionine, and D is UUA, UCA for leucine. Now, when you are looking at the choices, whenever we start with U, it primarily indicates it could be a stop codon. Hence, UAG and UGA are termed as stop codons or termination codons or nonsense codons, and predominantly these codons do not synthesize any amino acid. Instead of the amino acid, these codons, which are called stop codons, are involved in the release of release factors. The release factors in protein synthesis are designated as RF1, RF2, RF3. The release factors help in separating the ribosomes. They help in separating the small subunit and the large subunit. Liberating the polypeptide chain. And all the enzymes which are associated with protein synthesis. All the enzymes which are liberated, which are associated with protein synthesis. Hence, the formation of UAG and UGA as stop codons as release factors is at the end of chain termination in protein synthesis. In protein synthesis. The answer is B. Question number 80. Which one of the following pairs of nitrogen bases of nucleic acids is wrongly matched with the category mentioned against it? When you take A is thymine uracil pyrimidines, uracil cytosine pyrimidines, guanine adenine purines, adenine thymine or purines. We understand when you take thymine and uracil, they are commonly termed as pyrimidines. Same is the case with uracil and cytosine as pyrimidines with relation to the RNA and the DNA. And again, guanine and adenine are purines, whereas the last choice D, adenine thymine as purines, is wrong. Hence, the answer is D. Question number 81. Haploids are more suitable for mutation studies than diploids. This is because A. Haploids are reproductively more stable than diploids. B. Mutagens penetrate in haploids more effectively than diploids. C. Haploids are more abundant in nature than diploids. D. All mutations, whether dominant or recessive, are expressed in haploids. When you take haploid organisms have n number of chromosomes, and they are commonly seen in the case of neurospora. So, in neurospora, when you get haploid number of spores, the spore on multiplication gives rise to 8 spores, which are formed within the ascocarp. Ascocarp is a fruiting body, and this is the ascospores. The ascospores are 8 in number, and all the 8 ascospores are haploid in number. Now, whenever mutations occur, haploids always express themselves either in the dominant condition or as recessive condition. Whether it is dominant or recessive, always it is only haploid. Hence, they are used for mutation studies. The answer is D. Question number 82. What is not true for genetic code? A. It is nearly universal. B. It is degenerate. C. It is unambiguous. D. A codon in mRNA is read in a non-contiguous fashion. When you take the properties of a genetic code, the genetic code is universal. It occurs in right from E. coli to Homo sapiens. It is degenerate. Sometimes if the code is non-functional, it can code for multiple amino acids. it is unambiguous. It's very clear. Each codon is specific for a single amino acid. Whereas when you take choice D, a codon and an mRNA, when you have an mRNA on that you have a codon, is read in a non-contiguous fashion. Always codons are read in a contiguous fashion, that is, you have a sequence reading. You have 1, 2, 3 as a set, 4, 5, 6 as a set, 7, 8, 9 as a set, A, B, C, D, E, F, G, H, I, J, K, L. So, this method of reading is primarily termed as contiguous reading, whereas when you take choice number D is wrong, hence the answer is D. Question number 83. Removal of introns and joining of exons in a defined order in a transcription unit is called A. Tailing, B. Transformation, C. Capping, D. is splicing. So, when you take transcription, in transcription from the DNA, you have the formation of the mRNA. The mRNA molecule has introns, as well as exons. Exons are coding sequences. Introns are non-coding sequences. So, all the introns can be cut and removed. Introns can be cut and removed, and the exons can be joined. So, you have exon 1 combining with exon 2, and similarly exon to the power of 2n. All exons can be joined in a defined order. So, this method of cutting and pasting of the exons in a particular order, the term for this is termed as splicing. Splicing is a characteristic feature of transcription, and it occurs in the presence of an enzyme called spliceosome. Hence, the answer is D. Answer is D. Whereas, if something is added to the end of the mRNA, if X is added to the end of the mRNA, this process is called capping. If mRNA is changed, it is termed as transformation. And splicing is a term which is used for, in simple, removal of introns, sequencing, editing and adding of the exons, and whereby you have a sort of a transcription unit. The transcription unit is able to again synthesize the sort of an amino acids. So, you have a transcription apparatus getting ready for translation. The answer is D. The semi-conservative replication of DNA was first demonstrated in, the choices are A. E. coli, B. Streptococcus pneumonia, C. Salmonella typhimurium, and D is Drosophila melanogaster. Whenever we take semi-conservative replication, it has been demonstrated only in Escherichia coli because E. coli is easy to culture, E. coli's DNA is circular, E. coli has been well sequenced, again it is radioactive, and again it can multiply every 20 seconds. Hence, it is easy to gauge the life cycle of Escherichia coli. So, whenever we are dealing with semi-conservative replication, the model organism is E. coli. The model organism as studied by Messelson and Stahl is E. coli. Answer is A. '85. Whose experiments cracked the DNA and discovered univocally that the genetic code is triplet. Whose experiment cracked the DNA and discovered univocally that a genetic code is a triplet? A. The choices Hershey and Chase. B. Morgan and Studevant. C. Beadle and Totem. D. Nirenberg and Mathai. Nirenberg and Mathai primarily discovered that in vitro studies, when under test tube conditions or in vitro conditions, you take synthetic nucleotides and support them by addition of GTP, ATP and other cofactors. The synthetic nucleotides could synthesize an artificial amino acid like phenylalanine. So, when they use the synthetic nucleotides, these synthetic nucleotides which are used in a test tube were normally poly AAA or poly UUU. So, these polymers are primarily termed as homopolymers. So, homopolymers with giving under cell-free system protein synthesis. Cell-free system protein synthesis has been studied by Nirenberg and Mathai. The answer is D. Question number 86. Select the 2 correct statements out of the 4 ABCD given below about lac-operon. Question number 1. Glucose or galactose may bind with a repressor and inactivate it. Question number 2. In the absence of lactose, the repressor binds with the operator region. Choice 3. The Z gene codes for permease. 4. This was elucidated by Francois Jacob and Jaco Monod. The correct statements are, among the choices, if you look, whenever a lactose is absent, the repressor will bind to the operator region. So, whenever lactose is absent, a repressor binds with an operator region. This is choice number B or choice number 2. Now, along with this, the fourth one, this concept has been proposed by Jacob and Monod. Hence, the C choice says that in the absence of a lactose, a repressor will go and bind to an operator, and this sort of a binding results. And the concept of binding has been proposed by Jacob and Monod. The correct answer is C. Question number 87. The one aspect which is not a salient feature of the genetic code. This is a negative question which says which is not a salient feature of the genetic code is A. Degenerate, B. Ambiguous, C. Universal, D. Specific. Normally, when you take a genetic code, a genetic code is non-ambiguous. That is, it is very, very clear. Each codon should specify a sort of an amino acid. Hence, the negative statement, that is statement number B, is the right answer. Answer is B. Question number 88. Satellite DNA is an useful tool in A. Organ transplantation, B. Sex determination, C. Forensic science, D. Genetic engineering. Whenever we take SAT-DNA, SAT-DNA has repeat sequences. Repeat sequences means in which the DNA is conserved. And because it is conserved, it is an useful marker DNA in terms of history. Thereby, SAT-DNA is an useful tool for studies on forensic science. The right answer is C. Question number 89. Which of the following does not follow the central dogma of molecular biology? A. P, B. Mucor, C. Chlamydomonas, D. HIV. When you take the central dogma of molecular biology, the central dogma of molecular biology always says that DNA makes RNA and RNA makes proteins. DNA makes RNA, RNA makes proteins. Sometimes in viruses, RNA can also go to form the sort of a RNA can form the DNA. Now, when you take the central dogma, this process is called transcription, and this process is termed as translation. In addition to this, DNA can also form the DNA. This is DNA synthesis. Now, this central dogma is an universal feature in all living organisms. It is seen in plants like pea, fungi like mucor, algae like Chlamydomonas. However, it is an exception in the case of the viruses. Now, viruses primarily are termed as biological puzzles. They are termed as biological puzzles because they exist in the transition between a living and a non-living being. Under non-living conditions, they are inert as chemicals. Under living, they are infective in a living host. While if you take HIV, virus, HIV, it can bring about change whereby the RNA again goes to form the DNA. Reverse determinism is seen even in the case of HIV. Hence, it is an exception to the central dogma, that is the flow of information from the DNA to RNA to proteins. The answer is D. Question number 90. Which one of the following also acts as a catalyst in a bacterial cell? The choices are A. 5-SR-RNA, B. SN-RNA, C. HN-RNA, D. 23-SR-RNA. Now, when you take the families of the RNAs, in the families of the RNAs, when you take the small ribosomal RNA, the small ribosomal RNA 23 acts both like a sort of a catalyst as well as it also functions in the form of an RNA. is the right answer, and the answer is D. Question number 91. Removal of RNA polymerase 3 from the nucleoplasm will affect the synthesis of choices. A is tRNA, B is hnRNA, heterogeneous RNA, C mRNA, D is rRNA. Now, if you take RNA polymerase 3 and if RNA polymerase 3 is removed from the nucleoplasm, you have the nucleus, nuclear membrane, nucleolus, and this is the nucleoplasm. If RNA polymerase is removed, then it can, because of this, the mRNA transcription can go wrong, and when the mRNA transcription goes wrong, it affects the synthesis of the tRNA. The tRNA has got an anticodon. So, if the RNA is removed, it can affect the mRNA. This in turn can affect the tRNA. Hence, the answer is A. Question 92. Which one of the following is not a part of the transcriptional unit in DNA? A. Inducer, B. Terminator, C. Promoter, D. A structural gene. When you take the DNA and you look at DNA as a transcription unit, all DNA molecules have inducers. They also have a terminator. Then they also have a promoter and a structural gene. A promoter, a structural gene, and a terminator are ideal components of the basic DNA, whereas an inducer is an exogenous gene or an exogenous molecule. An exogenous molecule is not a component of the transcription unit. The answer is A, that is, an inducer is not a part of the DNA, we are also sort of a transcriptional unit. Question number 93. If one strand of the DNA has a nitrogen base sequence of ATCTG, what would be the complementary RNA strand sequence? The choices are TTAGU, B is UAGAC, C is AACTG, and D is ATCGU. Obviously, because there is time, there is T, the opposite of T is U, hence the choice is B, that is UAGAC. We go to question number 94. Removal of introns and joining of exons in a defined order during transcription is called A. Looping, B. Inducing, C. Slicing, D. Splicing. When we take transcription, during the transcription of mRNA, if intervening sequences which are called introns are cut or they are removed, and expression sequences called exons are joined together so that they can express during gene expression. The term which is primarily used for this particular process is termed as splicing. So when you take splicing occurs on the mRNA. So if we have a DNA sequence and the DNA is made up of introns and exons, if this is A intron and this is B intron, we use restriction enzymes which can cut across here. Thereby, the intron piece is removed. And if this is an exon called 1 followed by another exon which is called 2, exon 1 can join with exon 2. So the process wherein introns are primarily removed and one exon portion joins with another exon portion, this process or this technique is termed as splicing. So splicing which occurs in transcription in the mRNA normally occurs after transcription. So it is a post transcription modification. So among the choices of A looping, B inducing, C slicing and D splicing, the correct answer is D, that is splicing. Question number 95. Read the following statements A to D. Question number A. In transcription, adenosine pairs with uracil. B. Regulation of lac operon by repressor is referred to as positive regulation. C. The human genome has 50,000 genes. D. Hemophilia is a sex-linked recessive trait. How many of the above statements are correct? Normally, when you take here, we look at choice number A. In transcription, adenosine pairs with uracil. That is a wrong statement. Then B. Regulation of lac operon by repressor is referred to as positive regulation. It is not positive regulation, it is negative regulation. Whereas when you take choice number C, the human genome has 50,000 genes. Yes. Then again D. Hemophilia is a sex-linked recessive disease. Hence the right choices, we have got two statements and the answer is A. Next we go to question number 96. Which of the following forms the basis for DNA fingerprinting? A. The relative proportions of the purines and pyrimidines in the DNA. B. SAT DNA occurring in highly repeated short DNA sequences. C. The relative differences in the DNA occurrence in the blood, saliva, skin. And D. The relative amount of DNA in the ridges and grooves of the fingerprints. When we take DNA fingerprinting, if you take the purines as well as the pyrimidines, they are only taken as standards for study. Whereas when you take the DNA, in the DNA we have satellite DNA. The SAT DNA occurs as highly repetitive, short sequences which are conserved. The highly repetitive short sequences which are conserved are the basis for DNA fingerprinting. while the relative differences of the DNA which occur in the blood, saliva and the skin is a secondary choice, and the relative amount of DNA in the ridges and grooves of the fingerprints is a non-obvious choice. Hence, the right answer is B. Question number 97 deals with the central dogma. The following shows an important concept of the genetic implication of DNA. One of the basic properties of the DNA is it follows the flow of information in the form of codons, anticodons and amino acids. Fill in the blanks A to C. We have the choice as DNA making A, it goes to form mRNA, and mRNA in turn forms the proteins in the presence of B, and it has been proteins, it says, has been proposed by C. The choices are, if you take A says A is translation, B is transcription and C is Chargaff. Whereas choice number B says A is transcription, B is translation and C is Francis. Choice C says A is translation, B is replication, C is Rosalind Franklin. Choice D says A is transcription, B is replication and C is Watson. Now, when you take then the basic choices, we understand A is always transcription, B is always translation, and the concept of central dogma, that is DNA making mRNA, mRNA making proteins, the entire concept has been proposed primarily by Francis Crick. So, Crick is responsible for the central dogma and the answer is B.